Hello and welcome friends. Today we will solve December 2019 questions from Unit 6 and plant developmental part from Unit 5. Okay, we will analyze all part B and part C questions from these two units. Clear? Let's begin. So the first question is, this is a part B question. It is, which one of the following plants has this combination of key plant traits? Sporophyte dominant in the life cycle, vascular tissue, lack of seeds. Okay, so friends, this question is from diversity in plants, basically from plant classification, which is from unit 9. Since we are dealing with plant part, so I have included this question in my video as well. Okay, so to explain this, I would like to show you one chart. So see, the plant kingdom is divided into vascular plants and non-vascular plants. Vascular plants means the ones which have xylem and phloem. Okay, as you all know, xylem is a water conducting element and phloem is the food conducting element. Okay, non-vascular plants include mosses and liverworts. Vascular plants are the ones which can produce seed and cannot produce seed. The one which cannot produce seed, they are ferns, horsetails, club mosses. Okay, the ones which can produce seed are angiosperm and gymnosperm. Gymnosperm can produce the seed, but they cannot form flowers. Okay, so gymnosperm have naked seeded plants. Sometimes the question asks, naked seeded plants are gymnosperms. Okay, the examples include pine trees and evergreens. Now, angiosperms are the flowering plants and can be divided into monocots and dicots. So, coming back to the question, what is asked? It is asked that the sporophyte is dominant in the life cycle. Vascular tissue is present, but the seed is not there. Okay, so vascular tissue is present. That means this will not be the case. It will not be mosses and liverworts. Okay, so we have to proceed until before the production of seeds, because the ones which produce the seeds are included under angiosperms and gymnosperms. So the ones which are vascular but does not produce seeds are included under ferns, horsetails and club mosses. Okay, and we tell it as pteridophytes. Okay, the ones which can produce the ones which have vascular tissues but does not produce seed, they are included under pteridophytes. Clear guys? So the mosses here, club mosses or the ferns, see here the difference is, this is mosses which do not have vascular tissue but club mosses have, but here in question the correct answer will be fern. See, mosses are non-vascular. They are included under Bryophyta. Okay, ferns are included under Pteridophyta, and cycads are gymnosperms. Gymnosperms have seeds, and monocots are under angiosperms. Clear? Now this classification is very important for CSIR NET and for any other life science entrance exam, because these are the common points of classification which we have to remember. Okay, let's proceed to the next question. So answer is 2. Now coming to the next question. Which one of the following reactions take place during the reduction phase of the Kelvin-Benson cycle? So I tell you that Kelvin-Benson cycle or Kelvin cycle is also known as C3 cycle, and the plants which exhibit this C3 cycle are known as C3 plants. Okay, the C3 cycle is completed in 3 steps. Okay, this is in photosynthesis chapter. Okay, and the last part was under plant classification. This is under photosynthesis chapter, so I'll explain you by showing one picture. So see, 3 steps are there here. The one is carbon fixation, also known as carboxylation. In carboxylation, what we see, this is the first step. In carboxylation, what we see that there is a carbon acceptor, there is a CO2 acceptor named as RUBP, ribulose 1,5 bisphosphate, which will accept the CO2 and it will form 3-phosphoglycerate. So number 1 is carboxylation, where RUBP will react with CO2 and it will form 3-phosphoglycerate. Okay. The second step is the reductional step. In the reductional step, what we see that this 3PGA or PGA are the same thing. So 3-phosphoglycerate will be converted to glyceraldehyde 3-phosphate via an intermediate. So I am writing the overall reaction where this 3PGA is converted to glyceraldehyde 3-phosphate. This is reductional step. Now regeneration means the carbon CO2 acceptor, which is RUBP, is regenerated. Okay, and resynthesis of RUBP. So from glyceraldehyde 3-phosphate we get RUBP back. This is regeneration step. Now come back to the question. What we see here, see, ribulose 1,5 bisphosphate will form 3-PGA. Now this is a carboxylation step. Okay, 1,3 bisphosphoglycerate to glyceraldehyde 3-phosphate. I told you there is an intermediary step, 1,3 bisphosphoglycerate to glyceraldehyde. This is a reductional step. Okay, so this is reductional step. Now dihydroxyacetone phosphate to fructose 1,6 bisphosphate. These are the intermediary steps. Okay, for the regeneration. Ribulose 5-phosphate to ribulose 1,5 bisphosphate, which is RUBP. Ribulose 1,5 bisphosphate. This is a regeneration step. Okay, and they are intermediary steps. When you will study the detail about Kelvin cycle, you will find these steps. Okay, so 2 is the correct answer. Let's proceed to the next question. So answer will be 2. Now this question is from Plant Development Unit 5, but since we are discussing plant biology, I have included this question as well. So see here, the question is, which one of the following plants has a bisporic 8-nucleate bipolar embryo sac development? Now I tell you one thing, that embryo sac development is one of the favorite topic for the entrance exams, for the life science entrance exams. So in no way we will skip it. So let's begin. Bisporic plant development. So again, to solve this, I'll show you a picture which will give you the basic idea. So see here, there are different types of embryo sac development. It is monosporic, it can be monosporic, it can be bisporic, it can be tetrasporic. Okay, now monosporic is 8-nucleate. Okay, bisporic can be 8-nucleate, tetrasporic you can see it can be 8-nucleate or 16-nucleate. Okay, so these kind of questions are very common. You have to learn the example. Monosporic example is Polygonum and Enothera, and bisporic example is Allium. Okay, this question is repeated. When we will solve other papers, you will see it. Tetrasporic 8-nucleate is Adoxa, 16 is Peperomia, again 16 is Pania, and another 16 is Drusa, and 8-nucleate is Fritillaria. So if you learn the examples, then this will help you. Okay, so here bisporic example what we saw is Allium. So let's get back to the question. Now again one picture more I have. You can see here, see, Polygonum is monosporic, Enothera is also monosporic. Enothera is 4-nucleate, Polygonum is 8-nucleate. Okay, both are monosporic. Allium is bisporic. Allium is bisporic. Now Adoxa, Plumbago, Pania, Peperomia, Drusa, Fritillaria and Plumbagella, they all are tetrasporic. So they can give you one example and you have to match. So let's get back to the question. Which one of the following plants has bisporic development? So it will be allium. So answer 4 will be correct. I hope it is clear to you. Now another question. While screening an EMS mutagenized population of a plant, a researcher identified a mutant which with reduced gibberellic acid sensitivity. Which one of the following proteins is most likely to be defective in this mutant? Okay. So in simple language, this question is asking that which protein will be mutated, which of the following protein will be mutated as a result of which gibberellin sensitivity is reduced. Okay, that means which of the protein will be mutated which is not showing gibberellin sensitivity. That means to solve this again, I'll show you one picture. So you can see here, just for the general idea, I have kept this picture. This means that gibberellin hormone represses a protein named as DELA. Okay, gibberellin represses DELA. And what does DELA do? DELA represses phytochrome interacting factor PIF3. Okay, that means gibberellin is activating PIF3 because gibberellin is inactivating DELA. That means indirectly PIF3 is activated. Okay, now another picture, look here, that whenever gibberellin is present, okay, whenever gibberellin is present, in that case DELA will be repressed. So now DELA cannot repress PIF3, so gibberellin will bind with PIF3 and it will show stem elongation. Okay, and the stem elongation condition, if you have studied sensory biology, you must be knowing it, that this is a condition in dark. Okay, GA level is very high in dark condition and stem elongation occurs at that time. But what happens in light? Now in light, what we see that when the dawn starts, okay, when a dawn starts, we see that gibberellin receptor, which is GID, gibberellin receptor level decreases. As a result of which gibberellin cannot bind to GID, as a result of which DELA cannot be repressed, so DELA is stabilized at that time. Okay, so when DELA is stabilized, DELA is repressing PIF3. So when DELA is repressing PIF3, PIF3 is then degraded. Okay, PIF3 is then phosphorylated. It is then phosphorylated, leading to its destruction by ubiquitination pathway. Okay, leading to its destruction by ubiquitin pathway. So when PIF3 is not there, then there will be no stem elongation. So remember, in light there is no stem elongation, in dark there is an excessive stem elongation. Okay, in dark GA level is high and GA reacts with PIF3 to elongate the stem. So let's get back to the question. We can see here that when PIF3 or phytochrome interacting factor when it is mutated, then gibberellin cannot show its response. Okay, so 3 is the correct answer. And one more important thing you need to remember, that I always tell you that you have to understand about the other options as well. So sucrose non-fermenting related kinase 2, they are used in abscisic acid pathway. They are required in abscisic acid signaling pathway. Okay, constitutive triple response. If you know hormone, which hormone shows triple response? Yes, it's ethylene. So they are required in ethylene signaling. Okay, now coronative insensitive one, this is required in jasmonic acid signaling. These kind of proteins we find in these signals. Okay, so coming to the next question, which is also very easy. Which one of the following parts of root is involved in perceiving gravity? Now we all know that Root is the site which grows towards the ground. Root shows geotropism, but the developmental root also have different regions. So let's look at the picture. So look here, the root, if we do the cross-sectional area of the root, it has a maturation zone, elongation zone, distal elongation zone, and the meristematic zone. So meristematic zone means this is a region where from the division will take place. Okay, and this region have a kind of cell known as statocytes. Okay, this root cap region have a kind of cell known as statocytes. Okay, now the statocytes, so these statocytes are basically involved in the gravity perception. Okay, and these statocytes are located in the root cap region. As you can see here, these amyloblasts are present here in this picture in the root cap region, and these statocytes, they contain statoliths. Okay, these statoliths are starch filled amyloblastic organelles. These amyloblast organelles, they sediment at the lowest part of the cells. Okay, and helps the root to perceive the gravity. So these statocytes are the gravity perception cells, and they are present in the root cap. Okay, so let's go back to the question. So root cap is the region which has statocytes. So this is the region which perceives the gravity. Okay, so answer will be 3. Coming to the next question, another easy one. Vascular wilts are widespread and destructive plant diseases. The symptoms of this disease are primarily caused by the clogging of. Now clogging, I tell you that just for the idea, this you must be knowing when you were in class 11 or 12. Xylem are the vascular elements which helps in the transportation of water and minerals. Okay, xylem helps in the transportation of water and minerals, and these vessels are hollow. Okay, these vessels are hollow so that water can move upward without any resistance. Now phloem wells helps in the food transportation, and stomata helps in gaseous exchange, and hydatodes helps in guttation, if you know. Okay, the question is asking that vascular wills are widespread and destructive, and due to clogging of which kind of element, vessel element, do this kind of disease occur. So look at this picture. These are xylem vessels. Okay, these are xylem vessels which are hollow, that water can move from the root to the stem without any resistance. But due to several causes, say for example due to the infection by bacteria or fungus, these pores are being clogged. Okay, when these pores are being clogged, then the plants suffer from dehydration. Okay, and this dehydration leads to wilting of the plant. The dehydration and wilting, or vascular wilting, is due to the clogging of the xylem vessels, which helps in the transportation of water. So option 1 will be the correct answer. Coming to the next question, the tDNA region of a TL plasmid of Agrobacterium tumefaciens harbors two genes, X and Y. Mutation of gene X produces a rooty tumor, while mutation of gene Y produces shoots in the tumor. Based on the above information, which one of the following statement is correct? So it's saying that X gene produces a rooty tumor. That means when X gene is active, then that means when X gene is mutated, in that case only roots are being developed. Let me write for further clarification. Look here, just a moment. See, here it's saying that when X gene is mutated, so mutation in X, it produces roots or rooty tumor. That means which of the hormone we know produces root? We know that auxin helps in root formation. Okay, so in this case, Here root formation is there, that means root production is there. That means in the first case auxin production is normal, auxin gene is active, but there is no shoot production. Okay, mutation of, say for example, one gene does not produce shoot. So here we can say that the X is when mutated, it produces only root. Shoot is not produced. So here that means the gene for shoot production is mutated, because gene for root production is normal because root production is going on, but the gene for shoot production is mutated as there is no shoot formation in the first case. So we can say X is cytokinin, because here there is no shoot formation and the cytokinin is responsible for shoot formation. So when cytokinin is mutated, then there will be no shoot production and there will be only root formation. Okay. Now in the second case, when Y is mutated, when there is mutation in Y, in that case only shooty tumor develops. In this case, when Y is mutated, only shoot develops. That means the gene for shoot development is normal, but the root is not developing in the second case. So that means the gene for root is mutated here. That is why there is no root formation in the second case. That is because gene for root is mutated. So here the root formation gene will be the gene for auxin synthesis. Okay. So here Y will be auxin. Because when auxin is mutated, no root formation. When cytokinin is mutated, no shoot formation. Okay. So option C. So gene X encodes for cytokinin and Y encodes for auxin. Okay. Next question. Now let's see here. This is the question from sensory photobiology, and this is also a very easy one. Just a language, you may seem that this is very lengthy, but let's try to solve it. Consider the following facts. Chlorophyll absorbs more in the red region of the visible spectrum than in far red. Okay. The phytochrome photoreceptor P of plants occur in two interconvertible forms, which is PR and PFR. Okay. Where red light converts PR to PFR and far red light converts PFR to PR. Okay. So I'll explain you while I read. So remember, here it's saying that phytochrome, PR and PFR are two phytochromes. There are two interconvertible forms of phytochromes. When there is red light, PR is converted to PFR. Okay. When there is far red light, then PFR is converted to PR. It is saying that. Okay. PFR absorbs far red light and gets converted to PR. Third line. Growing of a sun plant under the canopy shed causes increased stem elongation. Now, in the previous question, in today's section only, we discussed that in the dark there is stem elongation. Okay, here it's saying that when the plants are grown under canopy means they are grown under shade. Okay, they are not getting the proper light, so they are under shade. That means I can say they are under dark, and under dark there is stem elongation. I told you. Gibberellin and phytochromes interact together to elongate the stem. Now, which one of the following combination of statement is correct for the plants growing under canopy? Under canopy means under shade, as compared to those growing above the canopy. So remember one thing here, one concept will apply, that during light, okay, during light, PAR will be converted to PFR. When the plant is getting proper light, PR is converted to PFR. And when they are under shade, okay, when it is under dark or when they are under shade, during that time we see that PFR is converted to PR. Okay, so we can say that if I write during light, red light will be more. So during light, red light will be more. Okay, and far red light will be less. Far red light will be less. So if I say ratio of R to FR, now for light, ratio of red light to far red light. And also remember one thing. So during light, red light is more and far red light is less. So I can say that the ratio of R red light to far red light is higher, because red light is more, so ratio will be higher. Okay. During night, what we see, during night red light is less. Okay. So red light is less, but far red light is more. So this is a general basic concept that if the denominator is higher, then the ratio becomes lower. So I can say that the ratio is lower because far red light is more during night. I can also relate it with shade or under the canopy. Okay. So ratio will be lower. Here ratio will be higher. Now this time we are considering with red and far red. Now what about PR and PFR? So when red light is more, then PFR will be more. See, PR will be converted to PFR in presence of red light. So when red light is more, then PFR will be more. Okay. So when red light is more, then in that case PFR will be more. Okay. And PFR will be more and PR will be less. So the ratio will be opposite. The ratio will be lower. Now for the dark one, understand. See, now again I write this one, PR to PFR. Now for if it's dark or if it's shade or if it's under the canopy, canopy is the vast spread of plants. So under the canopy what we see, that red light is less. Okay, red light is less, that means far red light is more. If far red light is more, then there will be PR concentration will be more. So if I write PR to PFR ratio for under the canopy or shade or darkness, we see that far red light is more. Far red light is more, that means the PR concentration will be higher. If PR is higher, PFR is lower, what will be the ratio? The ratio will be higher. If the numerator is higher, the ratio is higher. Okay. Now let's go to the question. So coming back to the question, what we concluded from the last explanation, look here. See, we found that under the canopy, the ratio of red light to far red light is lower, but PR to PFR is higher. Okay. So see, the red to far red ratio is lower. This is what we got. And PR to PFR ratio is higher, which is right. PFR inhibits stem elongation. Now one thing I told you, that during light, the stem elongation is inhibited. No stem elongation or inhibition of stem elongation. During light, there is inhibition of stem elongation. Now, and during light, what we see, PR is converted to PFR. So, during light, PFR is more. Try to understand, during light, PFR will be more. When PFR will be more, stem elongation is inhibited. Okay, so whatever reactions occurring in light is inhibiting the stem elongation. Right? So, 1 will be the correct answer. Now coming to the next question, which one of the following factors is known to be involved in the postponing? Now please try to focus on the question. Postponing programmed cell death in serial aluron until endosperm mobilization is complete. Now some people are there who will not read the question properly. They will see serial aluron layer. The answer will be gibberellin. Okay, because gibberellin shows aluron layer. Serial aluron layer formation. Okay, but here the question is postponing programmed cell death in serial aluron layer. Now serial aluron layer, what it does, it helps in the release of the amylase. Okay, now gibberellic acid helps in the release of the amylase from serial aluron layer. But if the which hormone will postpone the programmed cell death in serial aluron layer? So programmed cell death is an important task of serial alluron layer to release the alpha amylase. Okay, if now, so until endosperm mobilization is complete, the hormone which will postpone the process is abscisic acid. Okay, I already told you this is an inhibitory hormone. It has the antagonistic role of gibberellin. So postponing the role of gibberellic acid will be abscisic acid. Okay, the answer will be 2. So the question says that the plant produces cyanogenic glycosides to protect themselves from pathogens. Okay, a researcher has identified a variant of such plant that has higher level of cyanogenic glycoside, yet it is highly susceptible to specific fungal pathogen, because that means higher level of cyanogenic glycoside is there in that plant, still that plant is susceptible to fungal pathogen. How is it so? To interpret this counterintuitive observation, the researcher hypothesizes that the fungal pathogen has higher level of. So being cyanogenic glycoside present, still fungal can infect this plant. That means fungal is showing a different pathway. Okay, fungal is showing cyanogenic or cyanide resistant pathway, which is allowing the fungal to grow even in presence of cyanogenic glycoside. Okay, so one thing I would like to tell you, see, uh, now the question option says beta glucosidase activity. So remember, these are some enzymes given. Beta glucosidase activity helps in the formation of hydrogen cyanide. Okay, so one thing, the concept I would like to give you, that cyanogenic glycoside, they are stored in the non-toxic form, and when the plant parts are crushed by any pathogen, then only the cyanogenic glycoside produces the toxic substance, which is HCl. Okay, hydrogen cyanide, they are non-toxic, but when the plant parts are crushed by some pathogens or external agent, then they produce hydrogen cyanide. And this beta glucosidase, what do they do? Since this is a glycoside, so beta glucosidase break this glycoside to release HCl. So if HCl is released, in that case fungal will not grow, but here fungal is growing, so this will not be the enzyme. Okay, so I write here that cyanogenic glycosidase. Okay, cyanogenic glycoside, when the enzyme, this A enzyme will act, then in that case hydrogen cyanide will be produced. So when it is produced, then there will be no fungal growth, but here fungal growth is occurring. So this enzyme is not the correct option. Now coming to formamide hydrolase activity, now this is something new. That formamide, what does formamide do? Formamide hydrolase activity, it converts the cyanide to non-toxic compound formamide. So if cyanide is converted to non-toxic compound, that means it is creating the environment in which fungal can grow. Okay, so formamide can be one of the option. Formamide hydrolase enzyme activity can be one of the option. Cytochrome P450 enzyme, this also helps in biosynthesis of cyanogenic glycoside. This also helps in biosynthesis of cyanogenic glycoside. Now the last one, cyanide resistance alternative oxidase activity. So here what we see, now if you have studied the oxidative phosphorylation, there are four complex, 1, 2, 3 and 4. Okay, there are four complex, 1, 2, 3 and 4. 1, 3 and 4 are the major complex which releases the protons, and complex 5 pumps the protons. Okay, now there is one enzyme named as alternate oxidase. This alternate oxidase receives the electron from number 1, from complex 1. Okay, so that this 3, 4, 5 all are bypassed. As a result, from O2 water is produced. So complex 3, 4 and 5 all are bypassed. Okay, so here I have written the explanation. See, The alternative oxidase pathway only produces proton translocation at complex 1. Okay, 2, 3, 4 and 5 all will not work because alternative oxidase pathway works after complex 1. It receives the a proton from complex 1. There is no hydrogen pump, no ATP production over here. Complex 4 is inhibited by cyanide. So here we see that electron is received by alternative oxidase and it is not reaching the complex 4. Okay, it is bypassing 3 and 4 and 5. So we can say, and 4 is inhibited by cyanide. Okay, so I can say if cyanogenic glycosides are present, that means cyanide will be produced, and if cyanide is present, then there will be no ATP production. Okay, there will be like hindrance of the pathway, but we can see that cyanide is produced. Okay, cyanide is produced, but it is not reaching up to complex 4. Okay, it is completing its pathway in between by formation of alternate oxidase pathway, which is cyanide resistant pathway. Okay, so if the fungus fungus activate these two enzymes, okay, then they can still survive in the presence of high level of cyanogenic glycoside. In first case, if formamide hydrolase activity is there in the fungus, in that case they form the non-toxic formamide from cyanide, and in the fourth case they follow cyanide resistant pathway, means cyanide even if present they can show alternative oxidase pathway. Okay, so A and D are correct option. I'm sorry, B and D are correct option. So 4 is the correct answer. This is another part C question from secondary metabolites chapter, where you are being asked about like some metabolites will be given. You will be asked about their role and their source. Okay, so these type of questions have come several times in CSIR NET gate for the match of the column, and they are very easy to solve. So let's start. See, let's read. Several plants produced metabolites with important medicinal properties and have been extensively used in traditional medicine across the world. Many of these compounds can now be purified or synthesized and are used in modern medicine. Given below is a list of metabolites, their plant source and medicinal use. So we have to remember that digoxin, say the name, since this we all be knowing if we do not have so much of the detailed knowledge, that digoxin is produced from Digitalis purpurea, and this digoxin helps in cardiac arrest. Okay, next is salicin. So let's find out A3S. A3S is present in only this one. Okay, so this is the correct option. Still, we will go with the question first. So B, salicin. Salicin is produced from willow tree. Okay, and this helps in aspirin. Okay, this the role is the same as aspirin. Next is morphine. Now, morphine is produced from Papaver somniferum, and this is used as a narcotics. Next is artemisinin. Artemisinin is produced from Artemisia annua, and its role is anti-malarial. Okay, so even if you can match these two, I'll tell you the simplest way to solve this. Artemisinin, you know that D will be always be 1, so this and this can be true. Okay, digoxin, as the name says, it will be from Digitalis purpurea. So A will be 3, so A3 is present here and here. Okay, but this is not our option of selection. So this is the only option even if you don't know the detail about all. Okay, so this is a trick, and match the column have to be attempted every time. So answer will be 4. Now next, coming to plant diseases. Nowadays CSIR is targeting, even the gate is targeting about the plant diseases. They give some plant diseases and they ask us about the pathogen which causes disease. I'll make a short video also for this for you all. to remember about the disease and their key terms. Okay, so remember, whenever it's about blight, so blight is caused by bacteria. Okay, blight is caused by bacteria. Okay, so first let's know that starting with the rice blast. So rice, where is the name scientific term related to rice? Only here we can see is oryza. So rice blast is caused by Magna Forte oryzae. Okay, so B3 is present here and here. Next, coming to bacterial canker. So bacterial canker, here see that canker disease is caused by Pseudomonas syringae, PV syringae, and this is present in C2. And fire blight is caused by Erwinia amylovora, and powdery mildew is caused by Ersiphe cicachora cierum. Okay, this we have to remember. I'll make a short video on this for you to all remember about the plant diseases. Next, this is a question from photosynthesis. Now, C3 and C4 plants have been the favorite topic of the examiners. Let's begin. Which one of the following graphs best represents the net CO2 fixation of typical C3 and C4 plants under increasing CO2 concentration and saturating light? So it is asking us about the effect of CO2 concentration when the CO2 concentration is increased, as well as the saturating light on C3 and C4 plants. Okay, what is the effect of increasing CO2 concentration and saturating light? To solve this question, we must need to have the basic idea. What is the basic idea? That C4 saturates faster than C3. Okay, and the rate or the CO2 fixation, okay, the rate or the CO2 fixation, net CO2 fixation is less affected by the increase in CO2 concentration. Okay, because they saturate faster, so the net CO2 fixation is less affected by the increase in CO2 concentration. Let's see. So you can see here in this picture, in these four graphs, that C4 saturates faster than C3. So if you cannot see clearly, I write here this is C3 and this is C4. So here C3 is saturating faster. So this is not our answer. Here also C3 is saturating faster. This is not our answer. So answer can be from 3 and 4. Now see, look here. I changed the color. You can see here, observe here. Now, according to the first point, we have eliminated the first two options. Now, coming to the second point, net CO2 fixation is less affected by the increase in CO2 concentration. Here you can see that if the value of CO2 or this concentration of CO2, see, if the concentration of CO2 here is 100, then the net CO2 fixation is around 10. Okay, for C3 plants here we are studying, if the concentration of CO2 is 200, the net CO2 fixation is 20. So it's like, see, directly proportional. It's when the CO2 concentration is increased, C3 in C3 plants net CO2 fixation is also increased. Okay, for 300 it's around 35, for 400 it's around 40. So when the value of CO2 is increased, the net CO2 fixation is also increased. Let's go for C4 now. See, for C4 what we see, if the value of CO2 concentration, if the CO2 concentration is 100, it is 10. If it's sorry, so look here, if the value of, if this is 100, then the value of net CO2 fixation is 20. So it is increasing very faster. See, 200, for 200 it is around 40. Okay, it is around 40 or more than 40. So for 300, after that it saturates. So see, it saturates very faster, and even at low CO2 concentration, the C4 plants shows at least some net CO2, some photosynthate or some food is produced even when CO2 is very less. Okay, because they have the peculiar characteristics. They have the Kranz anatomy, which by which they sequester or they uh, capture the CO2 in the bundle sheet cells. Okay, so even at very low CO2 concentration, C4 plant can fix some amount of CO2, but C3 cannot show the net CO2 fixation at very low concentration of CO2. So what is the conclusion? That at very low CO2 concentration, C3 is not able to fix the CO2. Okay, but CO2 can. Okay, but CO2 can. So here this graphs become wrong because it's saying that C4 cannot fix at low concentration of CO2. At least here this graph is showing that C4 can fix but C3 cannot fix. So 3 is our correct option. I hope this question is clear to you. So remember that. Now next question, this is again from photosynthesis. Let's read now. C3 and C4 plant and CAM plants and C2 plants, at least one question each and every time you can expect. Okay, so please learn by heart about the comparison between C3, C4, CAM, photorespiration. Okay, now the table below shows photosynthetic type, temperature and sunlight intensity levels. Which of the following correctly matches the plant photosynthetic type with the temperature and sunlight condition in which photosynthetic rate per unit leaf area is maximum for that plant? So it is asking us that under what temperature and sunlight condition will the photosynthetic rate be maximum? Be maximum for C3 and C4 plants. Okay, this is our question. Now for C3 plants, now one thing we need to remember, that temperature is, now note, temperature is directly proportional to sunlight intensity. If the temperature is high, the sunlight intensity will also be high. Okay, so the temperature is high, sunlight intensity will be high. So high and low combination will not be the correct answer. Okay, now we know that C4 plants mainly grows in high temperature. Okay, C4 plants mainly they grow in high temperature. So B will be 1. So B will be 1, which is correct here and here. This 2 is wrong. C3 plant will not grow in high temperature. So if C3 is given 1, then it is wrong. So if A is 1 present somewhere, then it is wrong. Okay, not given. Now coming to that, if the temperature is high, then sunlight intensity is also high. So for C4 plant, if the temperature is high, then sunlight intensity cannot be moderate or low. This will also be high. Okay. So C4 plant will be B1P. So which is present in only the fourth option. And for C3 plants, they favor moderate temperature. They favors optimum temperature, which is around 25 to 40 degrees centigrade. Okay, this is the optimum temperature for C3 plants, which they favor. So if temperature moderate, I said since they are directly proportional, this will also be moderate. So A to Q. So fourth is our correct option. I hope this is clear. Now this is a question from plant hormones. We have to match the correct one. Now, auxin and gibberellin, cytokinin and brasinosteroids. Now, auxin, if you know that auxin and gibberellin, okay, they follow the ubiquitin proteasome pathway. They target their repressor proteins, or they tag their repressors by ubiquitin. When they are present, they tag their repressor by ubiquitin and degrade them by proteasome pathway. Okay, so and this auxins binds to the transmembrane receptor. I'm sorry, and gibberellin, they have soluble receptor. Okay, and cytokinin and racinosteroid. Cytokinin, if you remember, it follows the two component signaling pathway, like it is seen in bacterium quorum sensing. So cytokinin follows the two component histidine kinase pathway, two component signaling pathway, and the receptor for cytokinin and racinosteroid, they are the transmembrane receptor. So B will be A, which is here and here. Okay, and A will be B, which is here and here. Now again we go. Now I told you that by proteasome degradation pathway, and I told you that cytokine is a two component signaling pathway, involves several kinase, histidine kinase. Okay, so B, A and 1. Okay, so B, A and 1 is seen here, as well as soluble receptor, which is auxin and gibberellin. They degrade their repressor by proteasome pathway. Okay, so this is AB2, AB2 and BA1. This is the correct option. 2 is the correct option. Next, this question is again from photosynthesis. The following statements were made with the assumption that the concentration of 3-phosphoglycerate, is high in the chloroplast of an actively photosynthesizing leaf. Okay, what happens when 3-PGA is high? There will be high concentration of triose phosphate in the chloroplast. So 3-PGA, if it's high, this simply says that triose phosphate will be high. Okay, so this is again, this 3-PGA is kind of triose phosphate only. So 3-PGA is high, that means triose phosphate is high in the chloroplast. So A seems correct. So wherever A is correct, we can mark it. Okay, the activity of ADP, glucose pyrophosphorylase, will be inhibited. Okay, we'll come to this. Next, the carbon flow will be diverted from sucrose to starch. Okay, so if 3-PGA are too high and this amount of carbon skeleton is not required, then what will happen? Whatever will be required will be used up. Okay, so some will be used up and some will be diverted for the starch synthesis. Okay, all will be not required when there are high amount of carbon source, then some will be converted to starch, correct? Okay, next starch synthesis will be inhibited and carbon flow will be more towards sucrose synthesis. See, here it's saying that sucrose to starch will be formed, and here it is saying that starch synthesis will be inhibited. So either C or D can be correct because both are contradictory. One is saying that starch synthesis takes place, another is saying that starch synthesis will be inhibited. Both are contradictory. C and D cannot be together, and if C is there, D will not be there. Okay, so C and D are contradictory. We can eliminate the option like that. C is totally incorrect. I'm sorry, D is totally incorrect. Okay, now, so starch synthesis will not be inhibited. It will be promoted. Okay, there will be no more sucrose synthesis. So the extra glucose or extra the carbon skeleton will be diverted to starch synthesis, and the enzyme ADP glucose pyrophosphorylase, they will help in starch synthesis. Okay, so they will not be inhibited. So here A and C is the correct answer. Coming to the next question, which one of the following options related to plant growth and development? Now this is from unit 5. Is matched correctly? Leafy, regulator of genomic imprinting, xylogenesis, wood formation, MET1, inflorescence architecture, caspases, inhibition of apoptosis. Now the easiest one, if we will go with caspases, if you have studied apoptosis, you must be remembering that caspases helps in apoptosis. They are not the inhibitor. Actually, they are the ones which helps in apoptosis. So this is wrong. Now, What is the role of MET1? This MET1 is required for DNA methylation, if you remember, of three regions. DNA methylation of three regions in the MEA promoter in seeds. This is the extra information which you can write it down if you want, that they helps in DNA methylation of three regions in the MEA promoter in seeds. Next, xylogenesis is wood formation. This is perfectly correct. Okay, the xylem will ultimately help in the formation of wood, and this is known as xylogenesis. And leafy is a gene which is a floral meristem identity gene. Leafy is a floral meristem identity gene. They have no role in genomic imprinting. Identity gene. Okay, they have no role in genomic imprinting. Okay, so here B is the correct answer. Next question, this is from sensory photobiology. Which one of the following proteins regulates photomorphogenesis in plants by adding ubiquitin tags to a subset of nuclear proteins? So see here, I have to let you know the detail first. Now this is a very interesting question. It is asking that which of the protein adds the ubiquitin tags, okay, to the nuclear proteins so that photomorphogenesis is regulated. So let's see a picture. Here what we see that COP1 when it interacts with a protein named SPA. So COP1 when interacts with SPA, what happens? They add the ubiquitin tags to HY5 and HFR1. Okay, they add the ubiquitin tags to these proteins. So these proteins, what happens? These proteins are then degraded. Okay, multiple ubiquitin tags are then added, as a result of which these proteins are degraded. So when these proteins are degraded, there is no photomorphogenesis. So what we can say that this HY5 and HFR1, HY5 and HFR1, they helps in photomorphogenesis. Okay, so COP1 when it interacts with SPA, then this role is performed. So who is the main protein which is tagging with ubiquitin? COP1 helps in the tagging of HY5 and HFR1 with ubiquitin, and a SPA is acting as the activator of COP1. Now this happens in dark because in dark there is no photomorphogenesis. So this is a process which is seen in dark, where the HY5 and HFR1 are degraded. Now coming to the in presence of light, what happens? In presence of light, several other proteins like Cry1, PHYB, Phytochrome B, all these are what they are doing in presence of light. They are dissociating the interaction between COP1 and SPA. Okay, so this happens in this happens in light, where the interaction between the SPA and COP1 is dissociated by the proteins 5B, Cry1. Okay, as a result of which COP1 cannot activate itself, as a result of which this 2 cannot be ubiquitinated, and as a result of which they can show photomorphogenesis. So in summary, in the dark, COP1 it interacts with SPA, and when COP1 is activated, it adds the ubiquitin to HY5 and HFR1, as a result of which they are degraded and there is no photomorphogenesis. But in light, the interaction of COP1 and SPA is dissociated, so COP1 cannot add ubiquitin to HY5 and HFR1, as a result of which HY5 and HFR1 can show their response, that is photomorphogenesis. Okay, so the answer will be COP1. Now the last question, first committed precursor in the biosynthesis of chlorophyll is, the precursor is aminolevulinic acid.
Okay, remember this. Now, here I complete the December 2019 questions of plant biology, where I have included the system physiology plant unit 6, as well as I have included the plant development part, okay, from unit 5. So in short, I have described all the plant biology part of December 2019 in this video. Okay, thank you.